Okay, so with this video, I'm going to talk about resting membrane potential. And so as you've discussed in class, you've got a cell membrane. And so you've got things on the outside and you've got things on the inside. Now, along the membrane, there are many different things. But when it comes to membrane potential, we're going to focus on three things that are embedded within the membrane. Okay, and so we've got over here, right here, we've got the sodium leak channels. Right here, we've got the sodium-potassium ATPase pump, and then over here, we've got the potassium leak channels. Okay, now if you recall from class, the leak channels are hallways that are always open, and they allow ions to move down their gradient. Now, if you remember, hopefully you remember, you've got sodium, which is much more concentrated on the inside of the cell compared to the, much more concentrated on the outside of the cell compared to the inside. So it's always going to flow while this gradient exists. Sodium is going to flow into the cell. Okay. Now I want you to think for a minute, what is this going to do to membrane potential? Okay. And the answer is, and we're just going to do this in yellow, is that it's going to make membrane potential more positive / less negative. Okay. So in other words, this right here is a depolarizing movement. Okay, with sodium or with a positive ion coming in, that's going to depolarize the membrane if we just look at sodium moving in. Okay, next, and I'm going to skip over to the potassium leak channels. Potassium is much more concentrated on the inside of the cell compared to the outside, so that's why I've got big K plus on the inside, and so it wants to go down its gradient. And so we've got a positive ion leaving the inside of the cell going to the outside. And so this is actually, this is actually a hyperpolarizing or making membrane potential more negative. So I'm just going to put hyper here. All right. And then next we've got the sodium potassium ATPase pump. Well, the pump is actually what maintains membrane potential because what happens is this sodium that goes inside the cell, it's immediately pumped back out via the pump, okay? And so we have 3 sodium that leave the cell, and then but it also involves, wrong color. It also involves potassium, and so this potassium comes back into the cell, and so we've got 2 potassium coming back into the cell. So if we take into account the fact that we're pumping 3 positive ions out, there are a chart plus 3 out, but we're only bringing 2 in. This is also hyperpolarizing, okay? And so basically as soon as sodium enters the cell or potassium leaves the cell, it's pumped back in, but it's not an equal amount of charge. And so this helps maintain that negative membrane potential. And then the last thing that we've got is we've got these proteins. And so we've got all these proteins here. Okay, and they're gigantic, and I know I'm not drawing them big enough, but that's all right. Just think about if you had an elephant in your house, it's not going to be able to go through the door or through a hallway because it's too big. That's how proteins are, okay? And proteins are negatively charged. And so as a result, just having proteins on the inside of your cell is going to make membrane potential more negative, or in other words, it's hyperpolarizing. When we take all of these things together, we get a resting membrane potential in a neuron of approximately negative 60 millivolts. Okay. Now, if we got rid of the pump, I want you to think about what would happen. And if you think you know the answer of what would happen to membrane potential if we got rid of the pump, send me an email, let me know.